Hi there, welcome to Energy and Photosynthesis lecture video number 4. In today's lecture, we are going to be talking about what's called the C4 pathway and the CAM pathway. Before I want to talk about those, let's get a little bit cleared up of what they mean exactly or what they're related to so you have an idea before the lecture begins. So first off, I'm going to talk about C3 plants. C3 plants are plants, they're the ones we've already talked about. Plants that do the light reactions and then they do the Calvin cycle. So plants that only do the Calvin cycle are called C3 plants. All right. And that's because their first molecule that they produce, 3-PGA, in their Calvin cycle is a 3-carbon molecule. So they're the plants that are called C3 plants, or they do the C3 pathway strictly alone. All right. So C3 plants, they only do the Calvin cycle, C3 pathway. All right. They have what's called a mesophyll cell. That's where this is taking place, basically. They produce glucose. All right. These are some commonalities you're going to start seeing. C4 plants and CAM plants, well, there's some similarities to C3 plants. All of them do the Calvin cycle. Okay. C4 plants and CAM plants, they do an additional step that produces a 4-carbon molecule. So that's why they're called C4 plants. And CAM plants also produce a 4-carbon molecule, but they work differently than C4 plants. Okay, so we're going to talk about those differences in a second. Well, so what is unique? Well, most plants on the planet are C3 plants. Some plants have evolved. C4 and CAM plants are an evolutionary adaptation that allows these two types of plants to survive environments that a typical C3 plant would struggle in. So C3 plants, sure, they're most of the plants on the planet are C3 plants, but they cannot survive in every climate and in every environment. So plants have evolved to survive different places and different habitats. Let's look at some similarities. Mesophyll, mesophyll, mesophyll. They all have mesophyll cells. C4 plants have a bundle sheath cell also. These two do not. They all use rubisco, right there. They all produce glucose. They all do the Calvin cycle. Let's look at some differences, though. C4 and CAM plants have an additional enzyme called PEP carboxylase, right there and right there. CAM and C4 plants produce a 4-carbon molecule before they do the Calvin cycle, showing you those steps there. And CAM and C4 plants have this additional cell. They have mesophyll also, and they have this additional bundle sheath cell. All right, so we're going to talk about the differences and similarities and how these have adapted to their particular habitats. So let's talk about the C4 pathway. Why is it called the C4 pathway? Because some plants, as I showed you in the picture just a second ago, produce a 4-carbon molecule before they do the Calvin cycle. And the Calvin cycle is the C3 pathway. So C4 plants, let's give you a little backup information. C4 plants, they do a 4-carbon path, and they will then do the C3 pathway. They do both, right? What's unique about these plants? Well, they've evolved in a way where they have 2 different photosynthetic cells. Why is that a good thing for them? They can now compartmentalize. They can do certain jobs for the cell. These jobs allow them to become more efficient at doing certain things. So C4 plants are very efficient plants. We're going to talk about that and how they are so efficient. So what do? Well, let's first talk about stomata so you have an idea of what a C4 plant does and why it does it. Stomata are tiny little pores or holes usually on the underside of a leaf. What do? Well, they allow gas to be exchanged. Carbon dioxide needs to go in to the leaf, needs to go into the leaf. Why? Because plants need to do photosynthesis, they need carbon dioxide. Oxygen, when it's produced in photosynthesis, needs to leave the leaf. Too much oxygen, not good for these enzymes in here. Rubisco does not work when there's too much oxygen present. Does not work effectively the way we want it to work. Water is also released through the stomata. So oxygen and water leave stomata where carbon dioxide enters. Water leaving the stomata is what allows water to be, as it evaporates, that's how water leaves, it evaporates. It helps pull water up from the roots, up through the stem and into the leaves. So this evaporation of water is important for water to be pulled up towards the leaves. This oxygen needs to leave so that the rubisco enzyme does its job correctly. Okay, carbon dioxide enters so that photosynthesis can happen. All right, so this is a typical C3 plant. One big mesophyll cell, and everything's happening here. C4 plants, well, what do? Well, what happens is when the day gets warmer, okay, as the day gets warmer, they partially close this little stomata. Why does it partially close? Because as the day gets warmer, they will lose too much water from these tiny pores. So the evaporation of water it will happen at a very high rate during the daytime on warm days. So they don't close it completely, but they partially close their stomata. Okay. This partially closed stomata causes oxygen to get trapped in there along with this water also. Okay. This trapped oxygen creates problems. What does it do? And Rubisco does not work very well in the presence of a lot of oxygen. Okay. It will not fix carbon dioxide as well when there is a lot of oxygen present. So in the warmest part of the day, C4 plants, their stomata partially close, trapping in some of that oxygen, which now inhibits or prevents Rubisco from doing its job very well. So, how does a plant get Rubisco to work better, more efficiently when this oxygen is building up on the inside? Well, C4 plants have this extra pathway called the C4 pathway. They also have a mesophyll cell, just like this, but they have an additional bundle sheath cell. What's important about these two cells? Well, the mesophyll cell can store carbon dioxide for the plant to use in the Calvin cycle. How does it do this? It uses an enzyme called PEP carboxylase, and that fixes carbon dioxide even when carbon dioxide levels are low and oxygen levels are high. PEP carboxylase can store and hold on to carbon dioxide for the plant. So when the stomata partially close, and CO2 is not entering as well anymore, they can release the CO2 into the bundle sheath cell that will allow Rubisco and the Calvin cycle to continue. So what is fancy and important about these plants? They are very efficient. They need about half as much water as a C3 plant when producing the same amount of carbohydrates. So what do? They can store carbon dioxide and then they can release it into the Calvin cycle when it is needed later. And this is happening in the warmest part of the day when the stomata partially close. Okay, that's what's happening to C4 plants. So they are more efficient because they have an additional enzyme called PEP carboxylase, which allows them to hold on and store carbon dioxide as a 4-carbon molecule. Then it releases that carbon dioxide into the bundle sheath, and now Rubisco is not inhibited because it's now getting surrounded by the carbon dioxide that was released, and it allows it to continue to make its sugar. All right, so they use about half as much water as a C3 plant when producing the same amount of sugar. Corn and sugarcane are common plants that do this, basically. All right. So this is all the information I already told you a little while ago. Stomata in the leaves partially close to preserve water. CO2 levels decrease. Oxygen levels increase as a result of that. High oxygen and low CO2 prevent Rubisco from doing its job, the C3 pathway. So what do? These plants have that additional enzyme that's not inhibited when the stomata partially close. What does PEP carboxylase, that extra enzyme, do? It converts CO2 into a 4-carbon molecule and then releases that carbon dioxide into the other bundle sheath cell. And by releasing the carbon dioxide, Rubisco enzyme will no longer be inhibited and sugar production will continue. So when it gets really warm, C3 plants aren't making a lot of sugar. They're not able to do a lot of Calvin cycle. C4 plants, they are able to make more sugar. They can continue producing their sugar, whereas a C3 plant would have Rubisco inhibited. A C4 plant would not because of the CO2 that's getting released by the additional cell. So they have adapted. They have an adaptation. They can survive warmer climates and continue to produce sugar very efficiently. We grow a lot of corn in our country, and it makes perfect sense because it can feed a lot of animals and a lot of people. For livestock is eating a lot of corn in our country, so it makes perfect sense that we grow a lot of corn, considering how efficient it is to grow. All right, so it's showing you C3 compared to C4 plants. They both have Rubisco. They both do the Calvin cycle, but there's 2 cells, mesophyll and bundle sheath here for the C4 plant. So the stomata, they partially close in the warmer parts of the day to preserve water. And when that happens, carbon dioxide levels drop, oxygen levels increase. Increased oxygen is not good for rubisco, so it gets inhibited or prevented from functioning correctly. C4 plants can release the CO2 from that 4-carbon molecule that they produce into the extra cell, allowing rubisco to continue the Calvin cycle. So C4 plants, very efficient at producing sugar and glucose. The other pathway, called the CAM pathway. CAM pathway has some similarities to both of them, like they all do. They all have very similar. They're all very similar because they all do the Calvin cycle. And I'm showing you down here. CAM pathway is another type of adaptation. This occurs in cactus or cacti and pineapple. So what happens to these guys? Well, you got to think. A cactus is in the desert, and you're talking extreme dry environment, extremely warm, a lot of evaporation. So much evaporation that the plant could lose all of its water and die from complete dehydration. So what do CAM plants do? All right, what do CAM plants do? Well, they also produce a 4-carbon molecule, okay? They have a 4-carbon cycle, almost like a C4 plant cycle, but they do not have an additional cell. So C4 plants have an additional cell, CAM plants do not. They just have one giant mesophyll cell like the C3 plant. Okay, what do? Well, they're in the warmest part of the day, they need to preserve water, so their stomata don't partially close. Their stomata will completely close in the warmest part of the day. What do? So when their stomata are completely closed, they have no carbon dioxide entering because the stomata is completely closed, which means for them, the Calvin cycle would be completely inhibited in the daytime when the stomata is closed. But they need to survive. That would kill them. So how have they adapted, or what adaptation do they have for survival? Well, they have PEP carboxylase, that enzyme that fixes carbon into a 4-carbon molecule. They have it. What they do is at night, when the stomata are open, they take in and fix carbon dioxide into a 4-carbon molecule. So they're storing carbon dioxide at night. CO2 is stored at night. And then in the day, when the stomata completely close in the day, then they release that carbon dioxide that they're storing into the Calvin cycle. So it allows the plant to survive and do the Calvin cycle and make sugar for itself in those warmer parts of the day where the stomata are completely closed. Now, these plants, they're not completely they're not totally efficient. They are mainly designed, their adaptation isn't necessarily efficiency, their adaptation is survival. They can survive very warm, very dry climates without dying, basically, without completely dehydrating. So their stomata completely close in the daytime. No CO2 is entered. But when they're completely closed, they stored CO2 at night as a 4-carbon molecule. They release it in the daytime, and now rubisco can continue doing the Calvin cycle. All right. CAM plants do not have 2 cells like a C4 plant. Okay. They do not have 2 specialized cells. They are more like a C3 plant. They have 1 compartment. Okay. So these are an adaptation for hot and dry climates. So now that we've spoken about them, let's compare the two. This is a C4 plant and a CAM plant. They both have the enzyme PEP carboxylase. So that does the first step. PEP carboxylase does the first step for both of them. But what is unique about C4 plants? They have 2 compartments, and this allows them to have efficiency. C4 plants partially close their stomata in the day. CAM plants completely close their stomata in the day. CAM plants store CO2 at night and release it in the day for the Calvin cycle. All right. C4 plants can store carbon dioxide in that separate cell, that extra cell that they have, and then release it when it's needed to continue the Calvin cycle. So C4 plants, adaptation for efficiency in producing sugar with less water. CAM plants, adaptation for survival of very hot, dry climates. Showing you pictures of both again. I'm explaining the adaptations here for both of them, just like I mentioned previously. Okay. And once again, let's review all 3 together now that we've talked about all 3. Most plants, Calvin cycle, that's all they do. 1 major cell, the mesophyll cell, rubisco enzyme. CO2 enters, glucose comes out on the other end. All right. C4 plants, CO2 enters, mesophyll cell, PEP carboxylase, extra enzyme, 4 carbon molecules made and stored in this extra cell. Then it gets released when the stomata are partially closed during the daytime, and this allows the Calvin cycle to take place. Also has the rubisco enzyme. That's the term C4 plants, 2 compartments. Right? Cam plants, they also have PEP carboxylase. They make the 4-carbon compound first, CO2 enters, but at night they're storing carbon dioxide. At night when they're open, their stomata are open at night, closed completely in the day. So cam plants stomata completely closed in the day, but how do they continue doing the Calvin cycle so that they can survive? They release the CO2 that they stored from the evening. Rubisco can now fix it, fix carbon dioxide, and continue the Calvin cycle, making their sugar. What 3 things affect the rate or the speed of photosynthesis? They are light, carbon dioxide, and temperature. So if you shine light on a plant, the more light a plant has, the more intensity of light, it'll increase the rate of photosynthesis. But plants, they hit a maximum speed. There comes a point at which all of their chlorophyll, all the electrons have been energized or excited, and you can't energize or excite any more than all of them. So there's usually a max speed at which a plant can go. Any more light potentially could create heat and other problems. So those could be other issues that could be sorted by intense light. But light generally, intensity of light will increase the rate of photosynthesis and then hit a maximum speed or a peak. Okay. Carbon dioxide, same thing. Eventually, it can only fix so much carbon dioxide. There's only so much rubisco enzyme present. So once again, if you add carbon dioxide, the speed will increase, and then it'll hit a maximum speed and plateau. Temperature is a little different. As it heats up a little bit, the rate of photosynthesis goes up. It increases, it hits a peak, and then the rate starts to go down. Well, here's why. Temperature is one of the things that can denature the enzymes or proteins or change the shape of the proteins. It's from chapter 1, unit 1, in case you forgot. So since temperature can denature proteins, when it's too cold, the enzymes don't work so well, so photosynthesis is low. When it's too warm, the enzymes are denatured and they don't work as well, so photosynthesis rates are low. So there's an actual optimal temperature at which plants can do photosynthesis. Now, sugar, C6H12O6, all right? Sugar. This is the main product of photosynthesis, the main product. And to make this main product, light, enzymes, and chlorophyll need to be in place. Oxygen, this is a waste gas. This is a waste product of photosynthesis. So to the right of arrow, products, to the left of arrow, reactants. So in order for autotrophs, things that can make sugar and do photosynthesis, in order to make glucose, they need carbon. Where did that carbon come from? Follow the red. Carbon came from CO2. They need hydrogen. Hydrogen, follow the light blue, came from water. They need CO2. So follow the oxygen. That goes back to CO2. So oxygen comes from CO2, hydrogen comes from water, and carbon came from CO2. This oxygen, this is the oxygen gas that we breathe. Where did that come from? The breaking or splitting of water right there. Okay, here's our photosynthesis review. We'll stop there.